Okay, welcome back. Let's talk a little bit more about how DNA polymerase works and how it is doing its job well. So it needs to make an exact copy of the DNA parent strands that it's working from as a template. And while mutations happen, while mistakes happen, by and large, DNA polymerase does a pretty good job of making a very high fidelity copy or very close to exact copy of the parent strands it's working off of. There are multiple types of DNA polymerase, so we've talked about a few of them now in the previous video. We talked about DNA polymerase 3 as the major holoenzyme that's responsible for most of DNA replication. DNA polymerase 1 is the polymerase that's responsible for removing RNA primers and replacing those primers with DNA nucleotides. And three things that contribute to DNA polymerase doing this job well and efficiently are its processivity, fidelity, and exonuclease activity. So processivity refers to how many bases the polymerase adds before that polymerase falls off after it's completed its job. That sliding clamp component that latches onto the template strand is going to increase processivity because it prevents the DNA polymerase from falling off too early. DNA polymerase fidelity refers to its accuracy, how likely it is to add the correct nucleotide as it's adding new nucleotides to the growing strand. Whenever it incorporates the wrong base, its proofreading activity is going to increase fidelity because that incorrect base can be recognized and removed using 3' to 5' exonuclease activity. And exonuclease activity kind of gets its own category on its own because it is so important to maintaining the fidelity and processivity and functionality of DNA polymerase in general. So this is going to allow the removal of nucleotides that have been added either due to mistakes or by removing the RNA primer so that the final DNA double helix is only comprised of DNA nucleotides. All right, so proofreading activity, specifically removing the incorrect bases that have been accidentally added. All other things being equal, so no additional sources of stress on the cell like mutagen agents or things like that, DNA polymerase 1 and 3 has a relatively low mutation rate. So about one base in every 1 million bases that's been added in a growing nucleotide chain is by chance incorrect. This is the key source of spontaneous mutation and is responsible for a lot of the variability that we see between individuals of the same species. So this does happen, but it happens at a low enough rate that we don't have runaway mutation rates in lots of different organisms. Both DNA polymerase 1, which is responsible for replacing the RNA primers, and DNA polymerase 3, which is responsible for the primary DNA replication, these both have exonuclease activity that acts like a backspace key. So this proofreading activity to remove a base that's added to the 3' end of the previous nucleotide, it will remove that, insert the correct base, and then continue synthesizing the DNA fragment. So what this looks like, kind of in cartoon form, we have our template strand and our new or nascent strand. And DNA polymerase is moving in this direction. So it's reading 3' to 5', but it's adding new nucleotides in the 5' to 3' direction. In the second version of what's going on, we have the incorrect nucleotide added to the 3' end of the previous nucleotide. So again, we're moving in this direction. This is the incorrect base. So the DNA polymerase can pause and remove backspace and break that phosphodiester bond that was previously formed and remove this incorrect nucleotide so that the correct base can be added. So 3' to 5' exonuclease activity refers to this section right here, this directionality of moving from the 3' end towards the 5' end to remove that single base. The other type of exonuclease activity that's observed in DNA polymerases, specifically only in DNA polymerase 1, can move in the other direction. This has a particular relevance for dealing with those Okazaki fragments. So this is on the lagging strand. And whenever this RNA primer is encountered by DNA polymerase 1, this RNA primer needs to be removed. So our polymerase is still moving in this direction, reading 3' to 5', synthesizing 5' to 3'. It's going to encounter this RNA primer and remove it, exposing a gap here. This a DNA polymerase 1 will continue synthesizing, so that's what's shown with this arrow here, adding new nucleotides from the 5' to 3' direction and filling up that gap. So, to summarize, and this is another activity that you should do on your own before I show you the answer, I want you to fill out this table. So we have DNA polymerase 1 and DNA polymerase 3, and we've got different categories of activity. So this is polymerase activity, meaning that new nucleotides are being added. Does this happen in the 5' to 3' direction, the 3' to 5' direction? And then we have the two activities that we just talked about. So exonuclease activity, meaning that we're removing bases instead of adding them on. Polymerase adds things on. Exonuclease takes things away. So fill out this table. You can put in a simple yes or no in each of these boxes, and I'll go over the answer in just a second. So pause if you want a minute to work. Okay. So with this first category, we're talking about 5' to 3' polymerase activity for both DNA polymerase 1 and DNA polymerase 3. So remember, this one's responsible for filling in the gaps in the Okazaki fragment left behind between the Okazaki fragments on the lagging strand, and DNA polymerase 3 is responsible for most of DNA replication that occurs in the cells. So looking back at our sort of cartoons of DNA polymerization or DNA replication, our DNA polymerase 1 and 3 is going to read the template from the 3' to 5' direction and add new nucleotides, synthesizing from the 5' to 3' direction. So again, moving in this direction. So both DNA polymerase 1 and 3 are capable of doing this. This is the way that DNA replication happens in a global sense. So the answer to both of these is yes. Next, we have 3' to 5' polymerase activity. So this is suggesting that instead of moving this direction, the only thing different about this image is that it might occur in the opposite direction. So this is asking, does DNA polymerase 1 or 3 read from the 5' end to the 3' end, and does it synthesize from the 3' end to the 5' end, which is entirely backwards to what we just talked about in this case. And this is a case where neither of DNA polymerase 1 or 2 carry out this polymerase activity. It only ever occurs in this activity, this direction that was illustrated for this first category, this first question. This is never observed. The 3' to 5' polymerase activity is never observed. Next, we have differences in exonuclease activity. So first of all, we're looking at the 5' to 3' exonuclease activity. With this, we're asking, can our polymerase start from the 5' end and remove bases in the 5' to 3' direction? We know that DNA polymerase 1 can do this because this is what's required to remove those RNA primers between Okazaki fragments. So DNA polymerase 1 can do this, but this 5' to 3' exonuclease activity cannot be carried out by DNA polymerase 3. And then lastly, we have our last question. Can DNA polymerase 1 or 3 carry out 3' to 5' exonuclease activity? So focusing here now. And this is true for both of those types of enzymes. Both polymerases can perform that proofreading activity. So basically backspacing or removing an incorrectly added nucleotide. So starting from the 3' end and moving back toward the 5' end, 3' to 5' proofreading. This is a good table to practice. It's easy enough to fill in yes and no and kind of memorize where those answers go. But if you can work through each of these sort of visualizations that are shown for those different questions, that'll help sort of put in more concrete terms what these different polymerase activities are, what these different polymerase enzymes are capable of carrying out. Okay. This is another point for pause. We've gone through a bunch of different proteins that are involved in the synthesis of DNA through the process of DNA replication. So this is another good place to pause and see if you can figure out where all of the different enzymes go that we've talked about so far. So terms that you might consider using to fill in a template or a sort of fill in the blank diagram that looks like this are things like helicase, single strand binding proteins, DNA polymerase. There's only one polymerase shown on this image, so you should decide if this is DNA polymerase 3 or DNA polymerase 1. We also have RNA primers, leading and lagging strands, and you should also practice identifying where the template strand is relative to all of these different components. All right, so take your time to work on that. The answer is forthcoming. So on this diagram, I have essentially the same arrows and blanks that are shown on the previous side. We've got our helicase, which is indicated by this purple enzyme. This is going to move in this direction, unwinding that DNA double helix. Single-strand binding proteins, I'm only showing you one, but you can imagine that these are distributed all along everywhere we have single-stranded DNA. For simplicity, I'm only showing you one. We have the DNA polymerase 3, which is responsible for synthesizing the leading strand, which is shown here, and the lagging strand, which is shown here. I've labeled this Okazaki fragment. I would accept either answer for this type of diagram. We have our RNA primers, which are shown in red. This is required for any new nucleotide chain to start when in the context of DNA replication in a cell. And that RNA primer has to give us our 3' hydroxyl group in order for new nucleotides to be added, and so on and so forth in this direction. So one of the best ways to practice what you know, what you remember, to help solidify how all of these parts work together is to practice drawing out this replication fork structure. So there's a more detailed, sort of more graphically designed version of this replication fork on the following slide. Similar things are labeled in kind of a similar sort of orientation, just a different kind of perspective to look at. But as you're getting ready to study for the first exam, I would definitely practice drawing out this entire replication fork or structure. The better you can do this, the faster you can do this without kind of relying on a list of proteins or additional study aids, kind of like a cheat sheet, the better off you'll be in terms of thinking how all of these parts kind of come together. So this walks you through some steps that I would recommend doing to help solidify that information. Okay. So we've talked about initiation, we've talked about elongation. The last component of DNA replication is termination. So how do we land after we are finished replicating the DNA? This is going to vary between prokaryotes and eukaryotes, primarily because bacteria have a circular chromosome. So if you're trying to find a way to end kind of replicating around a circle, eventually you'll meet back up with where you started, and all you need to do is kind of heal that gap between the place that you started and the place that you ended up. And so termination is pretty straightforward in bacterial chromosomes, which are circular. On linear eukaryotic chromosomes, we have a more complicated process, of course. It's one of the kind of patterns with eukaryotes. Everything is more complicated. There's two main problems that occur with linear chromosomes. One problem is that you have two ends that aren't connected to one another. So how do you finish replicating at these two ends of the chromosome? And the other problem is that at the ends of these chromosomes, the kind of naked ends of the DNA resemble double-stranded breaks. And double-stranded breaks are recognized by a family of proteins involved in DNA repair mechanisms. And one of the things they can do is try and stick things together. So try and heal those double-stranded breaks by finding one end of one chromosome and maybe sticking it to another end of another chromosome, which is typically not good. The other thing that can happen with double-stranded breaks is they can be recognized by other exonuclease activity proteins that will essentially chomp and break up ends of chromosomes that are unprotected. So we have two things to kind of consider when we're thinking about the termination of DNA replication in eukaryotes. So if we're starting off with our representation of a linear chromosome, so pretend like this is a linear chromosome. This is one end, this is the other end. The first step in DNA replication is for helicase to unwind and denature the DNA, break those hydrogen bonds. And that gives us our two template strands. So we have our lagging strand template and our leading strand template. We're going to have primase, which is going to synthesize for us the RNA primers. And then those primase polymerase alpha, is going to be removed, leaving behind the starting point for our Okazaki fragments on this side and our leading strand synthesis on the other side. So once again, we're reading 3' to 5', 3' to 5' on both sides at the same time, and we're synthesizing in the 5' to 3' direction. Okay, so our next step is for DNA polymerase 3 to recognize the ends of these RNA primers and start synthesizing the new strand. So there's a caveat with drawing things out in 2 dimensions, which is shown here. So I'm showing you 3 enzymes just to demonstrate the point here, but remember that this is one large holoenzyme that carries out leading and lagging strand synthesis simultaneously. So for the sake of demonstration, I'm breaking some biology rules here. But what's happening ultimately is we have new strand synthesis on the leading and lagging strand at the same time. Once that's complete, those DNA polymerases are going to fall off, and we have our new strands with a few problems, right? So some of this contains RNA still. So these orange sections are our RNA primers. And then there's going to be some gaps between our Okazaki fragments that need to be fixed. So let's do that next. DNA polymerase 1 is going to use 5' to 3' exonuclease activity to remove the RNA primers. And then it's going to synthesize the nucleotides that go between the gaps of Okazaki fragments. But you'll notice that only one of these gaps was filled, this one in the middle, because DNA polymerase, just like DNA polymerase 3, requires a 3' end in order to start synthesizing and continue synthesizing until it reaches the point where it can stop. There's a problem over here on this side and on this side where there's no 3' end for DNA polymerase 1 to bind and synthesize the complement to this template component right here and right here. And so if this were to stop at this point, you'd have continual shortening of the replicated chromosome, which is also not very good. Okay, so what has to happen? After DNA ligase comes through and fixes any nicks that remain between the Okazaki fragments, we have to deal with these 5' ends that are left over after DNA replication is mostly complete. So once again, illustrating this a slightly different way, if you zoom in on this nucleotide right here, the 5' end is facing out, and there's no 3' hydroxyl group for the next nucleotide to come in and bind. So that polymerase activity can't happen. Okay. And like I said, these gaps at the ends of the chromosomes at these telomere regions would cause those chromosomes to shorten by the length of the RNA primer every round of replication that's carried out. So the answer to this biological question is telomerase. Telomerase is a large protein complex that is responsible for synthesizing the DNA at the ends of chromosomes. This is facilitated by repetitive DNA that's found at the ends of chromosomes in the telomere regions. They have lots of G repeats, many guanine repeats at the ends of the chromosomes. This is going to serve as a recognition site for telomerase to bind and start fixing this problem of replicating DNA at the ends of chromosomes. Telomerase itself is kind of similar to DNA polymerase in that it is composed of multiple subunits, and it is also a holoenzyme. So it contains all the subunits and cofactors that are needed in order to catalyze the telomerase activity. It's also known as a ribonucleoprotein. So ribonucleoprotein has a RNA component, so it has ribose containing nucleotides, and it has protein components. The RNA component is referred to as TERC, and that's illustrated in this picture down here. So TERC corresponds to this red RNA nucleotide component, and you can see a recognition site or a motif that's kind of spelled out in nucleotides on that RNA TERC component. The other component of the ribonucleoprotein is the protein component, or TERT. And this is something that is encoded as a gene somewhere in the genome, but in order for it to be functional, it has to be transcribed and then translated and then folded so that it can take on this telomerase protein structure. The entire holoenzyme, so everything that is encapsulated by these brackets, so TERT, TERC, and all of these accessory proteins are held together and function as a single unit. So the primary mechanism that allows you to take an RNA template and convert it into DNA is called reverse transcriptase activity. So we're taking the reverse of what typically happens in transcription, which is going from DNA to RNA, instead working backwards, going from RNA to DNA. This entire complex is stabilized by additional proteins. You don't need to know the names of these. I've just labeled them so that you can kind of see what they're called and relative to this entire picture of what telomerase is, so that it can kind of function as a unit. All right, so let's talk through the telomerase process. We're looking at the ends of chromosomes. So this is a little bit of a misleading figure. When we're working with DNA replication, DNA is not compacted into these chromosome structures necessarily. But to kind of give you an idea of where we're talking about relative to the long linear chromosome, we're focusing in on these very end pieces down here. And also up at the top, anything that's labeled in blue, basically. So at these telomere regions, you have telomere repeat sections. So everything that is kind of bracketed here is a telomere repeat. And I told you before that they are G-rich, so lots of guanine nucleotides added in these sections of the telomeres. These telomere repeat regions are recognized by telomerase. So telomerase, making use of its protein component and its RNA component, is going to recognize the ends of the chromosomes and attach itself, providing the RNA matching motif as a template so that reverse transcriptase activity can synthesize DNA using RNA as a template. So here we have our DNA that's synthesized using the RNA template. We've actually extended the length of the chromosome a little bit by the length of one of these RNA motifs. And this provides our 3' end that allows new DNA nucleotides to be added and fill in this remaining spot on the ends of chromosomes. So that's what's happened here. So DNA polymerase 1, which is, again, that enzyme that's really good at filling spaces, as long as it has a place to start, it's going to start at the 3' end, and it's going to start adding in these complementary bases. So for G, it's going to add a C. For G, it's going to add another C, a T, an A, and an A. Once this is complete, we're going to have the dissociation. So this telomerase protein is going to fall off, and we're left with a little bit of an overhang, so something that looks kind of like this. And that's going to be the subject of the next slide, kind of dealing with this thing that looks like a double-stranded DNA break. Okay? So, problem 1 is solved by telomerase. So our chromosomes are not shortening, they're getting a little bit longer with every round of DNA replication, facilitated by telomerase. The other problem, that kind of double-stranded break looking into the chromosome, is solved by Chalterin. Chalterins are, again, a multi-subunit protein complex, which is labeled here. So all these individual protein components comprise the Chalterin complex. These are going to take this double-stranded break-looking thing and loop it around so that it's protected from enzymes that are going to try to typically either stick double-stranded breaks together or start kind of gnawing away at the ends of the double-stranded break-looking chromosomes. So as far as what you need to know from this fairly complex diagram, we walked through the aspects and process of telomerase and how it adds on these little pieces of DNA to fill in gaps. So you should know that. But as far as Chalterins go, the information that's summarized in this text section is sufficient. So I'm not going to ask you to label anything over here. This is just to provide you some context for how this looks at the molecular level, at least diagrammatically. So focus here, have an answer for how the ends of chromosomes are stabilized. Okay? So one of the things that I'm sure many of you are thinking about is how telomerase and shortening of chromosomes is associated with aging and cancer and things like that. And if I just told you that every time cells divide and DNA replicates, the chromosome gets a little bit longer, that doesn't really match up with what you've learned about how aging and senescence happens. So what accounts for that difference? Let's talk about that a little bit more. So telomerase is an interesting kind of protein, ribonucleotide protein complex, because it's differentially expressed depending on the cell type and also the point in the cell's development. So what I mean by that is, if you think about cell differentiation in a complex multicellular organism like a human or a fly or a mouse, starting off with, at the point of fertilization, we have a bunch of these pluripotent stem cells. So stem cells that haven't differentiated but will eventually form all of those different types of tissues. So they follow through a bunch of different pathways. Some of them eventually differentiate to become the ectoderm, which gives rise to things like epidermis, neuronal cells, things like that. We have mesoderm, which is going to give rise to connective tissue, muscle cells, blood cells. Endoderm gives rise to the cells, for example, you find in your lungs or your pancreas. And then we also have germline cells, which give rise to eggs and sperm produced through the process of meiosis. So TERT, TERC, those RNA and protein components of telomerase, those are going to be expressed at a high level in these pluripotent stem cells, so prior to any differentiation. As these cells begin to differentiate, TERT, TERC expression is going to change depending on the type of cell that's going to be produced. So, for example, in the ectoderm lineage, we have cells that undergo lots and lots of cell division, so they divide many, many times. And in order for the cells that are destined for the epidermis to remain functional and to have telomeres that aren't shortened, that don't eventually erode away at genetic material, TERT, TERC is upregulated or remains upregulated in those high division rate cells. In other cell types, like nerve cells, muscle cells, blood cells, lung cells, pancreatic cells, things that are demonstrated on the slide, many other types of cells, these typically have a more limited lifespan, and they only go through a limited number of rounds of replication. So a rough estimate that actually varies quite a lot, but a ballpark estimate is cells in these categories typically divide between 30 and 50 times before they undergo senescence or cell death, essentially. You might have heard of apoptosis or apoptosis, programmed cell death. This is something that contributes to that process. In these endoderm and mesoderm cells and many ectoderm cell types, TERT expression is very low or completely turned off in the mature differentiated cells that fall into these categories. And this has something to do with, this actually leads to that observation of shortening telomeres that eventually start to erode away at DNA sequences that might contain genes. And when that starts happening, senescence is triggered, apoptosis is triggered, and those cells are eventually phased out. They eventually die. The other type of cell that's highlighted kind of in this green section where TERT, TERC expression remains high is in the germ cell lineage. And this makes sense. We would expect that the telomeres of chromosomes would be maintained or slightly elongated through telomerase activity in order to maintain gene continuity, I guess, or make sure that all of those genes are passed down to the next generation. So our telomeres are protected typically with meiotic cell division, and cells that divide really frequently have a lot longer lifespan compared to other cells like nerve cells, muscle cells, etc. So telomeres do shorten, but only in a large subset of cells. I won't say a small subset. Most of the cells are going to have the shortening of telomeres because they fall into this kind of purple category. Okay, so let's kind of look at how this process of TERT, TERC silencing happens. So here we're kind of sitting at the progenitor cell. These are the stem cells that were labeled on the previous slide. Telomerase activity is happening. It's making sure that these telomere regions don't shorten. So we're going to have an additional DNA section added here and here. But as soon as differentiation starts, the TERT expression and TERC expression is going to be silenced, which is what's indicated by this kind of stop line. Here we have a somatic cell. It could be many of these different types that are in this purple shaded region. Those somatic cells are going to go through maybe 30 to 50 rounds of cell division before the telomeres shorten to the point where genes are starting to be removed and those cells enter this apoptosis or senescence pathway. They break down, they're removed from the system before they can go rogue and cause problems, right? Okay, so this is what happens under non-cancerous situations. If we have a situation where TERT and TERC continue to be expressed and they're not inhibited, we have a cell that has no check on its lifespan. So basically, it's going to continue to go through cell division. Everything might be fine for, your 30 to 50 rounds of cell division, but because there's no check, because those genes at the ends of chromosomes continue to be protected because the telomeres continue to expand a little bit over time, this gets completely blown past, and we have something that's called malignant proliferation, which can contribute to tumor growth. So basically, these cells that have no regulation on telomerase activity become immortal, and they persist to overgrowth of cells. Okay? So because telomerase has some ties into how cancer cell proliferation can happen, there has been some focus on trying to develop chemotherapy drugs to target this particular enzyme so that it can be either silenced or reduced to reduce the overall growth of tumors or cell proliferation. So some things to think about. I'm not going to give you answers to these kinds of questions because these were intended to be more discussion-based questions for everyone to think about and talk amongst your groups. But it's important to kind of consider some of the benefits and consequences of using an approach like this, where you're targeting a holoenzyme, a very important enzyme that has a pretty clear purpose in the cell, in the organism. And if you're going to target that and cause it to be inactive or break it down in some way, there are likely going to be some off-target effects that you need to consider. So think about some benefits of the approach and how it could be appropriate for dealing specifically with cancer cells, and then think about some questions or concerns that you have as you think about all of the different cell types that might be influenced by the use of chemotherapy drugs that have kind of broad acting effects. They are specific in that they target a specific enzyme, but this enzyme does an important thing in lots of different cell types, and so as a result, the effects might be broader reaching. I know a lot of you are interested in medical tracks, so this is something that you can maybe find interesting. If you'd like to know more, I have a paper that's linked here. I believe you can access it on the lecture slides that are available to you, and you can read more about that. To get us set up and thinking about what's coming next class, we're going to be talking a little bit about the DNA replication mechanisms and stuff that we've talked about in this lecture, just so you have an opportunity to ask questions. But then we're going to transition into a more applied activity where we're going to take DNA replication mechanisms and look at how they've been used and repurposed to do molecular genetics. And there are a lot of different ways we could think about how DNA replication mechanisms could be leveraged to understand medical science. One is through the development of drugs that are often important for cancer-related research. So I mentioned telomerase. There's also cases where other components of DNA replication, everything from topoisomerase and polymerase to helicase and things like that, are targeted for potential cancer drug development. And you can think about, potential advantages and disadvantages of that, just like with telomerase. There's also instances where other kind of diseases can be investigated relative to controlling DNA synthesis of pathogenic bacteria. And there are a couple of papers linked here if you'd like to learn more about that. More detail than what we're going to cover in class and that you need to know for exams or anything like that, but definitely interesting. Other applications of DNA replication mechanisms that we've talked through come down to solving problems. So outside of maybe solving cancer as a problem, for example, we might have instances where DNA replication mechanisms can help trace the transmission of disease. We're actually going to talk about this one with the first case study. molecular procedures like PCR, something we'll talk about with the case study as well, are used for things like paternity testing and to understand how you go from DNA sequence to gene expression to phenotype. So RNA sequencing, for example, or proteomics all rely at the very most basic level on being able to take advantage of the information that's stored in the DNA sequence and then use that to move forward to other levels of the central dogma, essentially, in a biomedical context. So, some ideas to think about as you are reading through the preparation material for the case study on Thursday. Like I said, I'll give a kind of brief recap of DNA replication. We'll answer questions about that, and then we'll dive into case study 1. All right. If you would like to submit a test question for this, you're welcome to do that. I have the QR code here, as well as some assignments and reminders, so you can keep track of that. And we will reconvene on Thursday, because it's Tuesday today. All right. See you all then.